Very busy week this week with the news from Harmony. Of course, I'm thrilled that we are making progress at Spinogenix with SPG601 and providing hope with our modality, but I would also feel that this must be disappointing for the community and would love to hear your thoughts.
Yeah, thanks, Stella. I mean, I've been involved with the work with Zygel, going back to their first study, their first published report. And, I think anytime we involve hundreds of families impacted by Fragile X in clinical trials, and then have what's viewed as a negative study result or a real line of research that goes away, it's difficult. The nice thing about the cannabidiol gel was a really solid safety profile, and the administration of it, went well for a lot of families. I think some of the challenges there are, what was the preclinical justification for the mechanism? And some of my NIH work is actually focused on BD receptor signaling, where we've had NIH center grants that have investigated that with collaborators at Texas Southwestern and California Riverside. And we've not really been sure as a field what to do with CBD receptors, whether to block them or whether to agonize or support them. So this does provide, even as a negative result of some important scientific evidence, to tell us that just purely boosting CBD signaling is not having a really significant overall behavior effect. So I think even in a negative study, it's very useful information, but it's hard to hear. I mean, we know of families that have been in open label Zygel treatment for years. But I'm not overly, surprised by the result, but, it just tells us that other mechanisms we're studying, we need to have solid preclinical justification. And as you know, I really advocate for early human studies that are very quantitative. They look at what treatments are doing to brain activity and function before we do the huge studies, because we don't want to keep having these failures.
And so even with your experience, and obviously we just completed this study with SPG601 at Cincinnati with your guidance, exactly, doing these, it's very quantitative study, a lot of preclinical justification. Can you share a little bit more on just the enthusiasm based on kind of the path that you've helped us kind of progress and how this provides additional growth and where we're headed?
Yeah, I think, with the Spinogenix SPG601, first of all, the large conductance potassium channel is very well validated across a number of preclinical Fragile X studies. We know what the deficits are, which is different than going into all of the CBD cannabidiol work. We know that there's a deficit in signaling, so we're supporting that through use of SPG601. So that justification's there. And then, we took 10 men with Fragile X, gave them a single dose of SPG601 or placebo. We looked for brain electrical activity change and computer-based testing change, and we saw positive signals with both. So it's the smallest sample of patients ever where we've been able to reach statistically significant improvement with a single dose or even chronic dosing of a drug treatment, and that has me really excited. So that's the kind of de-risking I'm talking about of, hey, before we do big trials with SPG601 or any drug, can we have some early justification that shows, hey, this looks like it has some real potential? And 601 does, SPG 601 does. And those are using measures that are not responsive to placebo. That was a big issue with the CBD and the Zygel work. Big placebo response when clinicians, parents, and observers rate behavior. We all want patients to get better. That's why with SPG 601, we did EEG, we did computer-based testing. You don't get placebo responses there. Either the brain is functioning better, and with that, learning and memory are better based on computer testing, or it's not. And I think that's really the way to go. And I partnered with Spinogenix because you're open to that approach.
Well, yes, I'm very quantitative and I'm very excited about the path that we're headed on. And I'm thrilled, I'm beyond thrilled on the road ahead and just what we provide to the Fragile X community, having just completed our correspondence and communications with the FDA. We have the path kind of laid out as to next steps. And I guess we're really I'm looking very forward with working with you, Craig, in doing the next steps and providing other opportunities for the Fragile X community. And we're, again, very excited about this new mechanism, this new drug, and hopefully even with this setback, as you just shared, we do learn science. We continue to learn. Even a negative result is moving us in a positive direction on being able to then direct other studies and planning ahead. So we look forward to an incredible future for the field and also with our efforts.